Good afternoon to one and all. My seminar topic is neurotransmission. So before explaining about neurotransmission, I would like to briefly explain what is the neuron, the resting potential, the action potential, and propagation of action potential. Coming to introduction, neurotransmission is a term that has been derived from Latin terms. Transmissio means passage or crossing, and from transmitter that means send or let through. Neurotransmission is the process by which signaling molecules called neurotransmitters are released by the axon of a neuron, which is the presynaptic neuron, and the neurotransmitters bind to and react with the receptors and on the dendrites of the next neuron, which is also called as a postsynaptic neuron, which is located a short distance away. Neurotransmission is among the most important processes, and it is used by cells, tissues, organs, and organelle systems to communicate and carry out other processes vital for survival. Coming to the structure of neuron, by the way, the property by which all organisms respond to external stimulation is called irritability. Now, the nerve cells, also called as the neurons, have the main function of collection, conduction, and transmission of information. This information is nothing else but fast moving electrical impulses. Coming to the structure of the neuron, it contains a nucleus, and then there is a cell body. There are dendrites, which are short branches, and axon, which is the prominent extension. The stimulus coming from outside are collected by the dendrites, and the axon transmits this stimulus away from the cell body of the neuron. The cell body of the neuron is a metabolic center, synthesizes most materials for the cell. And in the diagram we can see that there is another structure, the node of Ranvier, and myelin sheath covers the axon, and the cells in this region are called the Schwann cells. That is the nucleus, and Schwann cells are responsible for producing the myelin sheath. This is the axon. Next, the resting potential. The membrane potential of a muscle or nerve that is unexcited is called as a resting potential. A voltage or an electrical potential difference exists between 2 points if there is a change in the charges between those 2 points. And when we talk about a neuron, the inside and the outside of the neuron have a difference in potential or voltage, and that is because of excess positive ions at one point and excess of negative ions at another point. The first experiment carried out to find voltage of the axon was on the giant axon of a squid, and that is shown in this diagram here. They used a voltmeter, connected electrodes to the inside and the outside. Here there is no potential difference because both electrodes are outside, but here one electrode is inside, other electrode is outside, so there is a potential difference of minus 70 millivolt, and they found the potential difference to be minus 70 millivolt, the inside being negative with respect to the outside. Now, membrane potential is not only present for nerve cells, it is present for all types of cells, and this can vary from between minus 15 millivolt to minus 100 millivolt. Moving over to the action potential. When a stimulus arrives at a neuron on the axon, the sodium channels open and the sodium ions diffuse into the cell. This reduces the membrane potential because there is a difference in charges, and this process is called depolarization. The depolarization can occur in two ways. The first way, by only a few millivolts. It will be between -70 and -60 millivolt, and when this happens, the membrane rapidly returns to its resting potential as soon as the stimulation has ceased. However, in the next case, if the depolarization occurs beyond the threshold, if it reaches -50 millivolt, then a new series of events is launched. Voltage gated sodium channels open. As a result, sodium ions freely diffuse into the cell because of the concentration and electric gradient. This causes the potential to reverse for a brief time, and the equilibrium for sodium ions, namely plus 40 millivolt, is reached. And when this happens, after 1 millisecond, the sodium channels spontaneously inactivate, and this blocks further influx of sodium ions. When this happens on one side, the potassium ions will move on another side. For this, the change in membrane potential caused by sodium influx triggers the opening of voltage gated potassium channels. This causes potassium ions to diffuse freely out of the cell. As a result, when the equilibrium potential of potassium ions is reached, which is -80 millivolt, the potassium channels will close and the axon will return to its resting state, and this entire series of events are referred to as action potential. So, looking back, what was said is, considering this as a membrane of the axon, there are positive charges outside, negative inside. If this is the sodium ion channel, when stimulus arrives, sodium channels will open and sodium ions will diffuse into the axon, and this will cause change in the charges. When the equilibrium is reached, that is plus 40 millivolt, sodium channels will close and it will cause potassium channels to open and potassium ions will diffuse out. When the equilibrium is reached for potassium channels, which is -80 millivolt, the potassium channels will close and this entire axon will go back to resting potential, and this entire process is called the action potential. I hope you understood the concept.